Yeah, I'm going to switch tracks a little bit and go from genetics over to hormones. And keep in mind, I'm not going to present a lot of data because we're just in the first couple of months of collecting data for this particular project. But I really want to go over the concepts of some of the work that we're doing. So I'm not going to go too deep into the data, but really to think about more conceptually what hormones play, what kind of role that they play in this whole dialogue about sex differences. And we have this really unique opportunity happening now clinically in trans health to examine some of these questions, not just within trans health and development of that particular domain, but how it relates back to sort of the larger landscape of what's happening in sex differences research. Before I get started, just really quickly, I have funding from a number of different sources, and I do some consulting for a parent media company. And so we will go over, some of my colleagues already presented the ideas behind sex differences in human research, why this might be an important topic, particularly in psychiatry and just more generally in cognitive and behavioral outcomes. I'm going to talk more specifically in this particular section about the importance of puberty and the idea of timing, that there's these specific developmental windows about how to think about development. And then we'll switch back into the transgender domain, really thinking about brain development as it relates to gender and sex. And lastly, to touch briefly on the specific role of hormones within the transgender context. So you guys will know by now, this is a really hotly debated topic, the whole idea of biology behind sex. And it's been debated for decades now of why this is important. And it's probably in response for a long time of we just neglected the idea of sex as a variable altogether. And so one kind of hypothesis is that it's really important to consider sex as we do anything, whether it's sociological or within medicine. And you can see in lay and pop psychology, but also in medicine, that it's something that's really easy to hang your hat on, this idea that there's a male version of everything and there's a female version of everything. And that same paradigm is very much true in psychology and psychiatry. So a lot of you probably heard of some of these books, including just one of our colleagues up at UCSF wrote The Female Brain, which was a blockbuster hit, about really defining that there's such a thing as a female archetype of a brain, and by corollary, behavior and interpersonal dynamics. And of course, there's a follow-up, the male brain. And then shortly after that, this whole idea of, hey, how are we interpreting gender and sex constructs overall in the way we approach brain development and in psychiatry? If you go and look at the literature and really try to determine, are there actually sex differences? There is a reasonable amount of data that suggests that if you just look at the phenotype, if you just look at behavior, some sex differences might emerge. So this is a meta-analysis that was done a number of years ago, but they looked at children's play behavior, identity, different cognitive and motor domains. And when you define effect size, it's the number of standard deviations typically. So a large effect size indicates there's sort of a large difference between one group and the other, and in this case, between the boys and the girls. And there's certain domains that really seems like there's a fairly robust effect size that separates by sex. So simple things like core identity, right? How do I identify as a boy or girl? And this is probably highly culturally dependent and time dependent, and I'm sure it's shifted quite a bit if they do that. Did the same sort of assessment more recently, where kind of non-binary gender identity has entered the picture more. But you can see the effect size, it's probably the most robust out of all these psychological domains of 11 to 13 standard deviations. But also things like play preference, who they choose as a peer for parallel or interactive play, as well as things like motor skills or activity levels still have a pretty robust difference between boys and girls. And in this instance, more in favor of boys for higher motor skills and activity level, and also for aggression. If you look further down the list, and I'm guessing it's a little too small, the print here, but if you look at the different cognitive and motor abilities, another really important thing to point out and to notice is that there's a number of domains that actually don't show any sex differences at all, right? And that might include things like spatial perception has a fairly small effect size, SAT mathematics has an effect size of 0.4, computational skills and verbal fluency, vocabulary, etc. People have also tried to look at this from the perspective of neuroanatomy. Now that we have MRI, let's not just look at the behavioral outputs, but let's also look at actual brain structure. And there is a series of study, one of the largest groups of neurodevelopmental images was done at UPenn, actually. And they scanned a whole bunch of kids as they came through the ER. And one of the things they looked at, obviously, was were there any differences in this population sample between boys and girls? And they came out with a number of articles that were, again, hotly debated once they hit the academic literature. And you can see by the responses in the lay media about finding difference between males and females. Is it actually a hardwired difference between male and female brains that can explain why men are better at met breeding? And the alternative hypothesis are people who say new insights into gendered brain wiring, or is it a perfect case study in neurosexism? So we have this task of trying to figure out what is actually going on here. Are there actually biological differences, and is that important? One of the best studies that's come out recently is a large sample, over 5,000 scans from the UK. And they looked at this in adult human brains. So the one I just showed you is from an adolescent population. But what they did is they took this sample of males, men and women in the overall population, and they tried to compare the differences again at the whole brain level. So if you look over on the left-hand side, these are the different components of how we measure the brain. One is total brain volume, and then there's subcomponents of gray matter volume and white matter volume. And an obvious sort of pattern that you can see is that, in general, men's brains are larger than women's brains. And this is something that's been replicated over and over in different data sets. And it makes sense. In general, men are taller, brain size roughly correlates to height to a certain degree. So it's not hugely surprising that there's a replicated separation in these global volume metrics. If you look at these subcomponents, so these are all smaller regions within the brain, it's not immediately as clear whether there's differences between the sex that follow along with the same differences you see in global brain volume. And these might be regions like the subcortical regions, the amygdala, the caudate, the putamen, thalamus, and other sorts of important temporal regions that are involved in a number of cognitive and behavioral functions. And part of this is how do we account for this difference, this sex difference in global brain volume, right? So one of the things that a lot of people don't take into consideration when they talk about sex differences in brain development is, if larger brains are bigger, anytime you look at a small structure within that, you're going to think, oh, well, amygdalas are bigger in men and it's smaller in women. And they might not necessarily be the case, because is it actually proportional to the differences that you're seeing at a global level, right? And so that's exactly what this study did, is they looked at mean differences without correcting for global brain volume. And it looks like throughout the brain, men have vastly larger volume, surface area compared to women. Interestingly, women, even if it's uncorrected, seem to have thicker cortices than men do in a number of regions that's not corrected. This middle column, if you go and correct for the fact that they have different larger total volume sizes, so on an individual case-by-case basis, they take out the variance associated with the larger volume, the differences shrink quite a bit. And what you're actually going to see now is there are still specific areas that do show sex differences, but now it's much more limited to targeted regions. And that's probably where we're going to put most of our interest now of thinking through sex differences. So a lot of the stuff disappeared in terms of surface area. What's interesting is also cortical thickness, the kind of thicker cortices in women compared to men, that was preserved even after correcting for this global volume. Another interesting thing from this particular study is that the range of variance, so how much does the smallest brain in men versus the largest brain, what is that span? That tends to persist. So men tend to have a much broader range of variance than women do. And that's across things like volume and surface area, and to a lesser degree in cortical thickness, which shows a more mixed picture. So you can see that when we talk about sex differences in brain development, it's a hairy topic, right? Like there's a lot of pitfalls that we can fall into. But it's an incredibly important topic. When I talked about really considering sex now in development, It's because, as my colleagues pointed out, Dr. Green, Dr. Weinberger, and others, that sex differences are clearly evident. If you're doing it from an epidemiological basis, we know the prevalence, how the course of progression, and also treatment. It's important to consider your patient, whether they're male or female, and to a certain degree. And it's also to address some of the shortcomings historically, where most of our research and clinical trials have predominantly included male subjects and tried to extrapolate those findings to both genders. We know in cardiovascular medicine that women experience myocardial infarctions differently from men. And if you're not accounting for sex, you may not be able to pick up some of those subtleties, right? So we want to avoid the situation where you develop skewed or biased diagnostic and treatment algorithms by creating these potentially incorrect conceptual frameworks. And one of the best ways to do that is really understand and leverage sex differences and trying to understand mechanistically what's happening in these differences and some of the observations that we're making. And this is particularly important, I'm a child psychiatrist in our field, because there is, again, this has been pointed out, a number of significant sex differences in child psychiatric disorders. So again, mentioned by others, in early childhood, so in these early neurodevelopmental disorders, so ADHD, learning disorder, ASD, it tends to manifest or present in kind of what we call early development or school age kids, and it tends to be really male predominant. There's an interesting phenomenon if you look at adolescent data, so as they're aging, there's an emergence now of female predominance in a number of disorders, like Dr. Green pointed out. So particularly in mood and anxiety disorders, MDD is major depressive disorder, and then also generalized anxiety disorder, separation anxiety. While these early neurodevelopmental disorders, the ADHD and ASD, tend to stay the same with male predominance, now, for some reason, these other mood and anxiety conditions, there seems to be a female predominance, specifically time-locked to adolescence. That's interesting because if you look at the epidemiological data from childhood, so this younger cohorts, there doesn't seem to be this sex difference. There doesn't seem to be a real variation between the number of boys who get depressed or the number of girls who gets depressed. That sex difference only emerges in this pubertal period. So why is that? We don't know. But if we think about it, probably to some extent, if we just think about sex, it's somehow mediating some of these processes, right? And we've heard some really great topics today about both genetic and epigenetic programming that happens very early in development. So all the way in utero, there's this priming or this programming that sets you up for different kinds of risk factors throughout life. And then also in utero, some of that is probably mediated by a hormonal milieu, and that varies probably between estrogen and testosterone profiles, as well as later developmental hormonal milieus, which I'll talk about now in more detail. And then the hypothesis is together all of these things in concert will drive changes in neuroanatomy and ultimately in the behavioral phenotypes that we're seeing in this very timed program of development. So I won't go really into this because it was covered in detail in sex chromosome aneuploidies, but I did want to point out, Dr. Green was looking at GWAS studies. So, we have these sex chromosome disorders with varying number of X and Y chromosomes, and these are really wide-ranging effects. So you would think, just from a genomic perspective, maybe it only influences the X and Y chromosome genes, but Dr. Rasnahan, the PNAS paper that Dr. Green showed, and then also we have data that we're putting out ourselves, what you can see is that it doesn't just influence this X chromosome. So this is the Turner syndrome versus XX female data. You can see these, from this plot, the differences in gene expression across this X chromosomes is the highest. It's the most different between groups. But anything above this black line shows that they're differentially expressed. So this is a hugely unexpected finding that came out in the PNAS paper and we replicated with RNA-seq, is that that's now extrapolated across the entire genome. So the fact that they have one less X chromosome, you're seeing the differences in this effect that's propagated through all of the autosomes. So, it's probably these regulatory effects through gene networks, but having some genetic influence that's gonna really underlie potentially broad-ranging sex differences, okay? One point, though, I want to make with genetics is that I think a lot of these effects will manifest fairly early in development. And hold on to that concept as we talk about hormones. So all of us in sex differences research, we often talk about these two main pillars of thinking about sex differences. One of genetics, which is really kind of the ugly stepsister. It hasn't gotten a lot of attention in sex differences research. Until recently, it's been getting a resurgence. But the bulk of what we've studied and what we look at in sex differences work is in hormone influence. And this stems all the way back from some really seminal animal work. I have a young son who says aminal, and I think I'm like picking up on his speech patterns. All right, so this is from early animal work back in the 1950s, where they did these incredible experimental paradigms. And what you do is you can take out the gonads from a particular, in this case they use guinea pigs, and if you expose in utero or prenatally to a different environment than what they were assigned, they're going to show much later in development completely different sex type behaviors. So in a very early hormonal interference or intervention can cause something, guinea pigs you can study because their lifetime is so much shorter than humans, but all the way later in adolescence and adulthood, they will show behaviors that are sex atypical from that one kind of exposure to a different hormone. So, obviously, and what the way they do behavioral phenotyping often in rodents is they'll look at mating behaviors. We obviously can't replicate that in humans, but it's this whole concept then, to riff off of what Dr. Weinberger was saying, of what's called an organizational and activational axis, or the organizational and activational hypothesis. And what happens is early in development, you're exposed to whether it's genetics or hormones, and your brain is rapidly sort of programming all of these different structures, all of these different functions in a way that's related to those two dimensions, right? And so it's organizing it in a way that might be different in kids who have more exposure to testosterone and to Y or XY karyotypes, and probably organizing it in a different way if you're exposed to more estrogen and XX karyotypes, right? And then the activational piece is based on this priming or based on this early organizational structure, certain events occur throughout your life that might activate or engage this priming in a certain way that will further diverge or further present more sex differences. And what we're particularly interested in is the activation that might occur from hormonal windows or hormonal periods that vary throughout life. And particularly, we're going to talk about puberty today, right? So we talked about this in early life. There's very few sex differences in affective disorders, but you'll see that there's a male predominance in these early developmental disorders. And then what's really interesting, and I'm going to focus here, is the adolescent period, where there's that increase in female risk and male increase in schizophrenia, which I won't talk about schizophrenia as much. It's, I think, a slightly different way of thinking about it than this particular model. So just going back, we're going to say, why is there's this switch is almost flipped, right? What happens in adolescence as you switch from this childhood pattern to this female predominant adolescent pattern? And sex steroids are a really great framework to think about this. They're highly prevalent throughout the brain, and there's profoundly different sex hormone profiles over development between males and females. And it happens in very specific times. In utero, there's a period of latency where they tend to be fairly similar in prepubertal boys and girls. And then in adolescence, again, hormonal profiles vastly diverge again. So we really need to understand these sex steroid signaling mechanisms in psychiatry. And when you think about it, we do have a challenge, though, in this from a scientific perspective. So if we're thinking about this organization piece and this activational piece that occurs later, it's essentially impossible to really discriminate between what is a pervasive organizational effect, right? And how do you discriminate that from what are the later hormonal activated effects? And that's because, for the most part, your assigned gender will match up with the genetic and hormonal experiences of that particular karyotype and or hormone profile. So I think what is a unique opportunity that we're looking at right now is trying to understand the specific influence of sex steroids in adolescence by working with a group of youth that identify as transgender. So in this particular study that we're doing now, we're taking a bunch of transgender youth, and we're specifically picking kids who are early in puberty. They're either prepubertal or they're just entering puberty. And now clinically, they're engaging in cross-sex hormone therapy at an earlier stage of puberty than in the past. So it creates a very specific paradigm now where kids who have an assigned gender, who have organizational programming that would be consistent with having that particular gender, whether male or female, are now entering a cross-sex pubertal milieu, hormonal milieu. So what that allows us to do is to really understand a little bit better what is actually happening in puberty that's specific to hormones versus what's already been primed or programmed from genetics and hormones from early in development. I'm going to cover very quickly, because there's almost no data, what we actually already know about transgender brain development and transgender health. And really, all the data essentially comes from Europe at this point. And that's because clinically, these are the countries where they've been ahead of the curve as far as treating kids and adolescents and adults who identify as transgender. And so there's a couple of small studies. The largest ones are from the Netherlands, but also smaller studies from Austria, Hungary, Spain, Sweden. One of the issues that I think we need to face as a field is almost all this data is in adults. There's really almost no data in adolescents right now. And if you guys are not familiar with the clinical paradigms for transgender healthcare, most of the treatments encompass cross-sex hormone therapy. So it would either be some variation of estrogen, progesterone, and or testosterone. And then sex reassignment surgery at later stages of puberty development. And if you look at the data, so some of these clinics have put out some studies, there's really limited differences. So if you're trying to find out, is there a brain region or a neural correlate, we call it, of gender, like can you distinguish the brain of someone who identifies as transgender versus not? There's very little that would indicate you can. So these are structural data that shows cortical thickness between men and women, and then also this group of male to female, basically transgender female adults. And overall, there's a couple of differences you might see in the occipital regions, and then also in the temporal sulcus, and also in some of the paracentral regions. So very minute or limited. In general, there's more similarities in pre-treated adult transgender females with peers of their assigned gender than there are to peers of their identified gender. And there's probably some very small specific neuroanatomical structures that will ultimately be correlated to gender identity. And this probably overlaps more largely with sex differences overall, but I'm hypothesizing it's going to be more specialized to gender. And very early on, back in the 90s, they looked at some histology samples and post-mortem brains. And some of the regions that you can identify are things like the sexually dimorphic nucleus of the preoptic area. This may not be meaningful to you guys, these names, but there's these tiny structures that are highly kind of sexually differentiated. The other one is the bed nucleus of the stria terminalis, and probably regions of the media amygdala. But these are all very tiny structures and that are highly populated with sex hormone receptors. So why is this a very timely conversation right now? What's happening clinically out in the clinical world is that there's rightfully a lot more attention now to what are the clinical interventions for younger adults or adolescents that identify as transgender. And what's happening is that medical interventions, which includes cross-sex hormone therapy, is optimizing for kids who really identify strongly as transgender to give them treatment earlier on so that they have better physiological outcomes later, right? So if you don't go through puberty, you bypass all these uncomfortable secondary sexual characteristics. And instead, you might have earlier on a pubertal development that's more aligned with your identified gender. The issue is the clinical data and the research data in this particular population of younger kids who are undergoing treatment now, it's really sparse. There's almost none. And I talked about some of those other studies, and they almost all look at kids who started treatment in later stages of puberty. So we rate puberty between Tanner 1 and Tanner 5, and almost all of those other studies, the average age is around Tanner 4. So they've almost completed puberty already by the time they're getting cross-sex hormone treatment. And there's a lot of variance still in how the pediatric endocrinology community is treating transgender individuals. And there's vast differences at the age of when they start treatment and when they consider stopping treatment, et cetera, et cetera. And it's important for us as neuroscientists to also think about that there's a scientific context to all of this. So we know even in cisgender individuals that there's a huge amount of change that happens in the brain during puberty. And that complicates our understanding of how to apply this in transgender health. So if you look at these differences, this is cisgender kids, is that when you look at large samples, this is from the NIH, you'll see that there are vast differences in the inflection point of how total brain volume is shaped across age between 6 and 20. And that happens more specifically in things like gray matter versus white matter. And girls might have earlier shaping, which correlates to the fact that they enter puberty earlier, et cetera. And unfortunately, there's probably only about one study that's been done in transgender adolescents that have looked really specifically at this, and this from a Netherlands group. And what they did is they exposed kids to this chemical odor that interfaces with one of those regions that I was talking about. And they found that, if you look at prepubertal kids, they don't have a brain response to that chemical signal that's any different from their assigned peers. But if you look at them after puberty, they tend to have responses that are more, at least the transgender female adolescents, they tend to have a response that's more in line with their female assigned peers than with the assigned male peers. So that would sort of emphasize the idea that puberty is a really critical concept here in the transgender population as well. Lastly, it's really looking at some of the treatment studies. So this is probably kind of the most influential literature out there that makes us really think about why it's important to think about this. And it's, again, really sparse literature. There's only been 3 longitudinal studies in adults and 1 in adolescents. And most of these are tiny sample sizes, so that's what we're trying to address in our particular work. It's looking at the differences between estrogen exposure and testosterone exposure as you go through this treatment. And if you look at some of the data, probably, again, you can see the sample sizes are quite small, groups of 8 or 6. And probably what's happening on a global scale is that testosterone increases volume, exposure to testosterone. So whether you're a cisgender male going through your natal puberty or if you're a transgender male going through cross-sex hormone treatment, it does seem that your brain volume in both the hypothalamus and in total brain volume tends to increase with treatment. And there seems to be a reverse effect with estrogen in the opposite direction. So this is similar data, just looking at that. So, one of my RAs here, Bobby, helped work on some of the early pilot data. We don't have a lot to show yet, but we're just looking at some of the initial time point 1 data of the kids that we have. And there's really very little differences right now prior to starting treatment. This is early pubertal group of kids in a number of psychological and cognitive measures. So at least at the baseline, we're not showing that there's a ton of differences between these 2 groups. And that includes both neurocognitive data and also our neuroimaging data. The sample sizes are too small, so I don't want you to put too much import into this particular, any of the images. But at least what it's kind of showing is consistent with what's seen in the larger literature, is that there's very few sex differences in prepubertal youth, transgender or cisgender. So here's the takeaway. For right now, there's really very limited evidence for neuroanatomical landmarks that show there's a gendered brain beyond maybe a few specific structures. So those structures may be important to really understand better and their function for how it relates to all these other things we're seeing, these sex differences we see in medicine. And there's no really clear diagnostic or defined neuroendophenotype for gender. I think that's really important too. There's also no clear adverse neuroanatomical or neurofunctional sequelae from cross-sex hormone therapy in adults. That's what we can glean from the literature as well. Going forward though, it's really critical, I think, for us to understand how do these differences in hormone profiles affect our long-term health trajectories. We know from cisgender literature that there's very specific developmental windows where your exposure to one hormone profile or the other can have a significant impact. And this is going to be really important, I think, also in advancing our ability to provide precision medicine, again, trans healthcare, but also for sex-specific determinants for health in cisgender populations as well. And in part, that's because we know that this interaction of sex differences in puberty, it touches a whole number of domains. So while I focus, and my particular interest is in brain development, There's good evidence to suggest that the timing of puberty is associated with differences in cardiovascular disease outcomes, diabetes, and a whole number of other diverse health outcomes in men and women, estrogens in cardiometabolic health, etc. So this is a really critical area to understand more. So I'm going to wrap up there. And just thank you to all of my collaborators, in particular Tandy and Steve, who are my peds endocrine clinician collaborators. Thank you.